Hello, good evening, everyone.
Good evening, sir. Good evening, sir.
I hope you are doing well.
God's grace to yourself.
I'm doing well.
We thank God.
Please, can you see my screen?
Yes, please.
All right, thank you.
You're welcome.
So we are talking about controlling microbial growth. So disinfection and sterilization. That's the first part. We shall come to the second part, which has to do with when disinfection or sterilization fail and there is infection, then you have to treat it within the host. So chemotherapy. So the classifications of disinfectants. We've talked about alcohols, aldehydes, phenols, and we are on halogens. So group 7 elements or group 17 elements are able to oxidize. That means they can remove electrons or they can add oxygen and increase the oxidation state. So once the enzymes or the proteins or other molecules are oxidized, they interfere with the activities within the cell. And that's how come these disinfectants are able to kill microorganisms. So for chlorine to react with water to form hypochlorous acid is very effective in killing microorganisms. That's why bleach, hypochlorite, and iodine are used for such purposes. But organic matter and other molecules like proteins, when they are in large concentrations, or the lipids, may inactivate these halogens. Some of them, like the iodine, can also be a stain, and nobody would like to use these disinfectants and end up staining the surfaces. They are also corrosive and can corrode the steel surface that are being applied to achieve disinfection. Then if you remember, we talked about classification of these disinfectants. So based on consistency, we have gases, we have liquids, we have the solid type or the powdered forms. So for the gases, we have formaldehyde gas, we have ethylene oxide, beta pipelatone, and ethylene oxide is very effective in killing microorganisms, including the spores. So it is a chemi-sterilant. And these are colorless. So once they are applied in the area or in the theater, they also have sweet smell, and one may not perceive them to be chemi-sterilants, but may assume that they are perfumes that have been applied in those rooms. So they have to be applied or sprayed with caution. And because they are very effective in penetrating substances, they are used to sterilize the heart-lung machines, the respirators, and even for domestic purposes where outbreaks of these infectious agents are suspected. They can also be used to sterilize glass, metal, paper, and other surfaces, or even for food products, but the concentration has to be reduced so that the individuals or the consumers will not end up harming themselves. And they are highly explosive. So they must be used with great caution. Formaldehyde gas is commonly used for fumigation in theaters and even in schools. And for beta propelatone, even reduced concentration to 0.2% is effective in killing microbial life and even inactivating viruses. So they can also be used for fumigation, and they are very effective in killing rapid spider. So the disinfectants will work when certain conditions are met. The activities of these disinfectants will increase when temperature is also raised because they are directly proportional in activity. If you increase the concentration of these disinfectants, you also achieve effective killing of the vegetative forms of the microorganisms up to a certain point. That is the optimum. So beyond the optimum, if you increase the concentration, you don't have any different outcome because that's a level enough to kill those organisms. But they may be inactivated by certain insulators like dead organic matter, in the case of proteins, fecal matter, or blood, or even non-organic substances like water, hard water, and some plastics that have trapped air. Time is needed to make these disinfectants work better. The longer the time of exposure, the better. And some of these will work against certain range of microorganisms. Others are effective against a wide range. So some of them target gram positives only and not gram negatives because gram negatives have the outer membrane in addition to the cell wall. So this outer membrane, which is rich in the lipopolysaccharides, also protects the cell wall from the action of these disinfectants. Same for non-enveloped viruses that are more susceptible to the action of these hypochlorites and glutaraldehydes compared to those that have envelopes. So these envelopes protect the viruses against the external environment. So at least these factors affect the effectiveness of the antimicrobial agents. So when you talk of antimicrobial agents, they could be the ones that are used externally on inanimate objects or surfaces, or as antiseptics, or the ones that are used within the host for treatment. So that's a general term for those that act against microorganisms. So the population size, if you have larger population size, require longer time to kill them. That's why for some medication, they are given over a period of time so that you can achieve effective clearance of those microorganisms. Same applies to when they are on the inanimate surfaces. You also need some time to clear if they are in large numbers. Some microorganisms may also be in the endospore state and are highly resistant to heat and other chemicals compared to those that are actively reproducing because they are in the vegetative forms. The concentration also affects the activity or the effectiveness of these antimicrobial agents. So increasing the concentration will also enhance the activity. But in some cases, if you increase the concentration and they evaporate rapidly. You may end up losing them before they can work or react with the organisms. So it's better to dilute them to a certain concentration before they can be effective. That's why we dilute ethanol to about 70 to 80% before we apply them for effective killing of these microorganisms. The time is also important for the effectiveness of the action of these antimicrobial agents. And as you recall, high temperature also enhances the activity of chemical reactions. And the environment within which these antimicrobial agents are working should also be considered. Is it acidic or alkaline environment? So heat in an acidic environment will enable the antimicrobial agent to be more effective than in the alkaline environment. Also, you can have some insulators like organic matter that may protect the microorganism from the action of these antimicrobial agents, or even when you apply heat. So for this case, you need to extend the time of exposure if such an environment is encountered. So these antimicrobial agents either kill or inhibit the growth by targeting the cell membranes. You know, the membranes contain phospholipids. So once the lipids get dissolved with these antimicrobial agents, or the proteins get denatured, they expose the cells and its components, and any material can go into the cell to destroy it or affect the cellular activities or metabolic activities. And once the proteins get destroyed, that means most of these activities cannot go on, because protein is what makes the genes get expressed. The genes control the activities of the cell by being expressed as proteins. So once the proteins are not there or destroyed, the organisms cannot use these enzymes to transport molecules or to gain support for its survival. Some of them also damage the DNA or the RNA, that's the nucleic acids. Once the DNA or RNA is destroyed, the organism cannot replicate, reproduce, because before they can reproduce, they need to double the chromosomes by replication. But if the DNA, which is to be doubled, is destroyed, there's nothing to replicate. So we cannot have the genes expressed to bring about the metabolic functions within those organisms. So that's for disinfection and sterilization. We come to the second part where there is infection established and we want to control the microbial growth within the host. So disinfection and sterilization occur out of the host. Control of the microbial infection occurs within the host or the system. And this is an in vivo activity happening within a living organism. In this case, we are looking at the living host. So these chemotherapeutic agents kill or inhibit the microorganisms. We are talking about microorganisms because this is microbiology. But in general, chemotherapy is the use of chemicals to treat or to manage conditions or diseases. But we are talking about microbiology, so we limit ourselves to infections and the diseases. And this chemical agent that is used to kill or inhibit the organism should not also kill the host. So selective toxicity should be considered where the host is not harmed, but the microorganism is killed or harmed. So antimicrobial agents could be antibiotics, which are chemicals produced by other microorganisms against other microorganisms to kill or inhibit them. Or they could be synthetic agents, where they are not produced by other microorganisms, but are produced by formulating or combining other compounds or active ingredients to achieve killing of other microorganisms. So for antibiotics, the first discovery was by a happy accident where Alexander Fleming with his team left a plate with Staphylococcus growing on it on the bench. So when they returned from vacation, they saw fungus, mold, growing around a portion of the medium, the agar, that they had left on the bench. But there was a clear zone around the mold that prevented the Staphylococcus from growing. But there were some parts where there was no mold growing, but the Staphylococcus was growing rapidly. So it made them suspect that there could be some chemicals or compounds within this mold that is preventing the bacteria, Staphylococcus, from growing. So they later identified this mold as penicillium. That's why they named the compound they extracted from this that was able to kill the or inhibit the growth of the Staphylococcus as penicillin. So since then, scientists have been trying to extract different compounds or agents from or chemicals from different microorganisms to see if they can kill others. So mostly they consider the normal flora and see if they can extract certain chemicals from them to inhibit the growth of others. So the selection of these agents for killing or inhibiting the growth of microorganisms has to do with the target organism. So if the organism is naturally resistant to the agent, why would you go ahead to prescribe? Or if the chemical targets only gram-negative, why would you prescribe it against gram-positive? So the target organism should be considered. Again, the side effect. If it is effective in killing the organisms, but will cause harm to the host, that means safety concerns have to be considered. And how long they can stay within the system when they are administered. You need to have them stay in the system for a long time so that they can clear in residual population. So how they are also metabolized within the system to bring about their desired effect. The selectivity, is it broad spectrum or narrow spectrum in their activity? So usually for most cases, the clinicians may decide to start with broad spectrum. If the test result is not ready, then when it's ready, then they narrow down to the specific ones for effective management of the condition. So these antibiotics can be classified based on the activity. Are they killing? So bactericidal. Are they inhibiting the growth But hydrostatic, are they targeting the cell or the cell membrane to lyse or break them? So bacteriolytic. They can also be classified based on a spectrum of activity, narrow or broad, in killing or inhibiting these organisms, or based on how they act. Some of them will inhibit the cell wall synthesis. Some of them will affect the permeability and the support or the protection that the cell membrane will offer, so that instead of being semi-permeable, they allow everything to enter and then destroy the cell. Some of these antibiotics also inhibit protein synthesis. So remember, proteins are very essential for enzymatic activities for expression of the genes. So once this is prevented or blocked, that means the organism is going to suffer. For DNA or RNA synthesis, these nucleic acids are essential for the replication to occur before the cells can divide to bring about reproduction, and also for the RNA to be used to make proteins. So all these can be inhibited and the cells will not be able to multiply or grow. So the choice of these antibiotics depends on which pathway or target the clinician wants to use to manage the condition. Sometimes the clinician may prescribe 2 different antibiotics that will target different mechanisms of action, but usually not the same mechanism of action. So the cider, the static, or the lytic, depend on how the clinician wants to kill or inhibit them, but usually they administer the cider ones, the ones that kill, in individuals that have poor immune system or compromised immune system. Because for those ones, if you don't kill organisms, they can overgrow and outnumber the strength of the immune system. So it's better to kill them so that the numbers become low or reduced. Then the bacteriostatic ones, they inhibit the growth of the organisms. So once the immune system is strong, and then you stabilize the microbial load, the immune system can take over and then control them. So for the antibiotics that target the bacterial cell wall synthesis and the permeability of the cell membrane, we have the types that have double rings. We call them the beta-lactam ring. So penicillins have these rings. Carbapenems have these rings. Cephalosporins also have these rings. That's why they are referred to as beta-lactams. So for the cephalosporins, cefalexin, cefrozyme, cefotaxime, ceftriaxone, carbapenems, meropenem, imipenem, meropenem, etapenem. But the clinicians who administer these antibiotics, we have first generation, second generation, third, and fourth. They will not start from the fourth generation. They start with the first, second. If they fail, then they go to the next class, third, before the last option, so that we can safeguard these antibiotics and prevent or reduce resistance. So penicillin, we have the narrow spectrum type and then the broad spectrum penicillins. So usually if the clinician is starting treatment and the confirmation is not yet ready, they may start with the broad spectrum, and then once the organism is identified and then the test is ready, they may switch to the actual antibiotic to use. For those that inhibit protein synthesis, we have the aminoglycosides, the microlites. These are the antibiotics naturally. They occur, and these are produced from other microorganisms. So streptomycin, neomycin, kanamycin, gentamicin. Microlites can also be able to inhibit protein synthesis. But we have the semi-synthetic type that can be derived from kanamycin to make amikacin. So the clinician may decide to prescribe erythromycin depending on the organism, or going for streptomycin depending on the organism, but they all inhibit protein synthesis. The ones that target DNA and RNA synthesis, the nucleic acids, those are the fluoroquinolones, the ciprofloxacin, the norfloxacin, the moxifloxacin, levofloxacin, the ones that end with floxacin. They all target DNA, RNA synthesis. But the antibiotics alone cannot work well unless you detect early. Sometimes the patients or the clients will come with the late stage of the condition. In that case, treatment may become very difficult, especially when organs are damaged or tissues are damaged. So early detection is key for effective treatment. And once you detect, you need to select the right antibiotic for better management. That's why they test in the lab which antibiotics can be effective before the clinician will prescribe. And need to optimize the dosage so that we reduce resistance that will be circulating within the population. Once we don't work on the dosage and we use suboptimal concentrations, a time will come the organisms may become resistant and subsequent treatments may fail. So the antimicrobial sensitivity or susceptibility test is used to measure how well the antibiotic or antimicrobial agent can inhibit the microorganisms from growing in the test tube or in the lab. So once we know that this antibiotic will work in the test tube experiment, then the clinician will be very confident to prescribe it. And there are two ways to go about it: the dilution method or the disk diffusion method. So this test will control the use of antibiotics so that the clinician will not be doing trial and error. Try this today, tomorrow try that, because this can increase the chances of long hospital stays or mortality rates. But if you know this mute, but if the test is performed in the lab and the clinician knows the ones that are working and the ones that are not working, he or she will go in for the right ones for initiation of treatment. So the first option, the dilation method, is quantitative. So here we want to know the minimum concentration that is in the antibiotic that will be able to inhibit or kill the microorganisms. And here we use antibiotics of different concentrations. So if it's Cipro can be prepared, and then we have different test tubes with different concentrations of the Cipro. So from low to high concentration in different tubes. and then we put the bacteria in the different test tubes that have different concentrations of the antibiotic. So the very low antibiotic will not be able to kill the microorganism, but the very high levels will suppress or kill them. So once the lab person identifies the bacteria, they pick about 4 or 5 of them and then put in a liquid medium, a broth. Then they mix gently, have the uniform or resultant solution. Then they prepare different concentrations of the antibiotic. So once they have different concentrations of the antibiotic, so here you have 0.5 milligrams per liter, 1 milligram per liter, 2 milligrams per liter, 4 milligrams per liter, up to 256 milligrams per liter. Then they introduce the organisms that they had put in the broth after incubation. They put them into the respective test tubes that have different concentration of the antibiotic, and they set up controls. So here there is no antibiotics, so 0. Here there is no bacteria. There is no antibiotic also here. So because there's no bacteria, there will be no growth here. That's why you are seeing this as red and not green. For this, there is no antibiotic, but there is microorganism or there's bacterium. That's why you see green, a big growth. So once you have these different concentrations and the organism bacterium introduced in them, then they culture on medium to see which ones will grow. So you pick petri dish, you plate here, you plate this one also on a different petri dish, you label to the last tube. Then you incubate to see if there will be growth. So the one that had no antibiotic, it means that the organisms are free to grow. So there will be growth. You see green, low concentration, there will be growth. Low concentration, there will be growth. It gets to a point where there is no growth on the petri dish. So we come back to check the test tube to know the concentration of that antibiotic that did not allow the organisms to grow. So that becomes the minimum. So beyond the minimum, no organism will grow. So 64, if there's no growth in 64, obviously there will be no growth in 128. But 64 is less than 128. So the 64, that was the starting point to inhibit the growth of these microorganisms, becomes the minimum inhibitory concentration, MIC. So now the clinician knows that if he wants to treat such condition, 64 will be ideal. If he decides to increase to 128, it will work all right. 256 will work all right. But what if the toxic levels will harm the patient? So why not come to the minimum and then probably spread it over certain number of days or weeks to clear all the bacteria? So this is a dilation method for checking. Yes, militant.
Sir, please, I don't understand the broad dilution method.
So this is used to measure the lowest concentration of the antibiotic and prevent the growth of the bacteria. No, please mute. So you prepare different concentrations of your antibiotic. So let's say Cipro. In different tubes, you have different concentrations of the same Cipro. This is more than this. This is reduced by 2. This is reduced by 2. Reduced by half, sorry. So as you reduce the concentrations, you introduce the bacteria into them. Because they have different concentrations, you don't expect the same growth rate. So for this one, there is no antibiotic. And remember, antibiotics kill or inhibit growth of organisms. So if there's no antibiotic, there will be growth. This one, there is antibiotic, but the concentration is very low, 0.5 milligram per liter. So the organisms can survive in the presence of this antibiotic. So they are resistant. Same for this one, which is low, there will be growth because they are resistant. These two, the organisms can survive. But it gets to 32, they survive at this concentration. 64, they don't survive. That means 64 is high enough to kill or inhibit the bacteria. That means the clinician doesn't need to prescribe this 32 milligrams per liter, this concentration to kill, because the minimum concentration that will kill the organisms are 64. If the clinician decides to increase to 128, it will still kill because 64 is killing, so increasing it will kill. But increasing it may be toxic to the client or the patient. So why not come to the 64 that was able to suppress the growth? That's why you're not seeing green. The green represents the colors of the isolate. That's why you are seeing green here. So here there is no growth. There's no growth because the concentration is increased. Here there's no growth because concentration is increased further. Here there was no bacteria, so we don't expect growth. Millicent, are you okay? So the minimum concentration is 64 because 64 milligrams per liter was the minimum compared to the others that were able to suppress the growth to inhibit the organisms from growing. Yes.
Sir, please, how about the number 4? When you were talking, you said you pick a sample, then we spread it on the plate. Yes, please. Can you enlighten? Yeah.
So these are coming from, so after adding the organisms from here, from number 2, you have prepared your concentration. So you add the bacteria from number 2 into the respective test tubes. Are you following?
Yeah, yes, please.
So after doing that, you pick different petri dishes. So here you can have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 different petri dishes, or you can take one petri dish and divide into 4, so 4 quadrants. So for 4 quadrants, you can plate 1, 2, 3, 4, 1, 2, 3, 4. Pick another one, you divide into 4, you plate the next 4. It depends on the lab resources. So they will grow on the petri dishes and also incubate this one. Remember, this one has the antibiotics and the isolates. Once you grow them also here, after overnight incubation, is the same outcome. If they were able to grow in this test tube, you can plate them or subculture, we say subculture, we subculture onto another medium and there will be growth. If they were killed here, there's nothing here. So once you subculture, there will be no growth here. Are you following?
Yes, sir. Please, I'm following.
Okay. So what you are seeing here, number 5, what you are seeing here is a representation of the growth that you are going to see on the Petri dishes. So if you see green here, it means you are going to see growth. If you see green here, you are going to see growth when you plate. Or you can incubate the test tubes alone and use stability to check the growth of these microorganisms. In our previous lecture, remember, we said we can use stability. So you will see clear zones for these ones because there's no growth. For these ones, they'll become turbid. But I can still plate and see if there will be colonies or not. For this one, there will be colonies because there's no antibiotic. For this one, there will be colonies because the concentration is low. It gets to a point where the minimum concentration is able to inhibit, and that is the 64. Is it clear now?
Yes, sir. So please, it means you can either use the plate or the stability, which is like the
Yes,
cube.
yes. Any is
Okay.
fine. It depends on the resources of the lab. Some for research purpose, they can do the two because they want to see, and then they have a scale for measuring the turbidity, the McFarland scale, to assess the turbidity level if it's 0.5, if it's 1. That's for research purpose. But for clinical examination, you want to see isolates or not, growth or no growth. All right. So let's move on. If there's no question, is there any other question, please? All right. So the next option is to use the disk diffusion test or the Kirby-Bauer test. Here, you don't do dilutions like this type. And this is what they commonly do in the hospitals. The hospitals, they don't usually do the dilution approach. It requires a lot of time. So here, and again, let me say that these are also done mostly when they want to come up with, please mute. Yeah. Mostly the pharmaceutical companies also use this approach to formulate the right concentrations or the strengths of their products. So for this diffusion method, these are having paper disk of known antibiotic concentration. So if it's gentamicin, the concentration is known and incorporated into a paper disk. And the same medium that will support the growth of the microorganism is presented, and the organisms are plated on the medium evenly. So you spread them evenly on the medium. So the entire medium gets the organism spread. And one way to do is to emulsify the test organism in saline and then spread them uniformly with sterile cotton swab. And you place your antibiotics that have known concentrations on the medium that has your test organism. Then you incubate. The antibiotics from the paper will diffuse onto the medium, and this medium, remember, has the bacteria. So there will be exposure or interaction between the antibiotic and then the organism. And once you incubate, we expect growth because there's medium nutrients. But the antibiotics are also expected to inhibit the growth of these bacteria. So if they're able to inhibit them, they will form a clear zone around the disk. And this is what we refer to as a zone of inhibition. And this is relative to how susceptible or sensitive the bacterium is to that antibiotic. So here we have different antibiotics that are placed within the dispenser. So you can have the gentamicin here, vancomycin here, erythromycin here, arranged with the concentrations written or inscribed on them. You have your petri dish with the medium. You plate your isolate first, spread them evenly onto the medium before you dispense the antibiotics. Or you can use the forceps to pick them manually and then drop them onto the petri dish. So usually 6 or 5 discs per petri dish, then space them out. Then use the forceps, which is sterile, to depress them so that they don't fall off or they'll have firm grip onto the medium. Then incubate at this point. Once you incubate, the antibiotics within the disc will diffuse onto the medium. And as the organisms grow, the antibiotics will then get exposed to them and inhibit their growth. So once you bring them out, you will see a clear zone around the disc, meaning the organisms were not able to survive in the presence of that concentration of the antibiotic. Once you see no zone around it or small zone, it means that they're able to survive or adapt to a certain limit. So you measure the diameter of this zone. So from this end across the disc to the other end in millimeters. You measure for each of these antibiotics that you have used, and then you compare. You have a standard. The chart is there. You compare, and what you have on the chart, that's a break point. If what you have and what you measured, if what you measured is more than or equal to what you have on the chart, that means the zone of inhibition is good enough and the organism is sensitive. If what you measured, please mute. If what you measured is less than the breakpoint on the chart, assuming you measured 19 and the breakpoint for Cipro is 20 or 21, because the 19 is less than the 21, the organism will be able to survive and it is resistant. So that's how we assess the sensitivity of. Please mute. Regina, please.
There.
Yes.
Please, when you were starting with the disc diffusion, please can you go back to the previous one?
This one?
Yes. So you see the first one, you said you first impregnate the disc with the antibiotic before you introduce the culture on it. Right.
So when you buy the disc, it already has the antibiotics formulated and incorporated into the disc.
Oh, okay.
You buy from the manufacturer or the pharmaceutical company, they have this disc with the known concentration already. So you just place the disc onto the medium.
Okay. Thank you.
But with this, the dilution method, you have the antibiotics in a bottle. So you reconstitute, you have it in solution, and then you prepare your concentrations. I hope it's clear.
Yes, please.
Right.
Thank you.
Okay. So somebody was asking, let me see. Rahimatu is asking if this is the same as the discovery of penicillin. So this idea led them to use this method. So Kemboa, they built on the idea of inhibition of the staphylococcus by the penicillium. So this is the idea that they built on. Yes. Okay, so two scientists were named after this, Kirby and Mboa. And for gentamicin, you see there is growth around the disc. So if you measure this diameter, you may get about 7 or 6 millimeters. Usually when you have growth around the disc, the disc diameter is 6 millimeters, so you may record 6 or 7. But this one, you are seeing a large clear zone for fosomycin. That means this concentration of fosomycin is able to inhibit the growth of this particular microorganism or bacterium to a larger extent. But we cannot just use this clear zone size to conclude that it is resistant unless we measure the diameter. So from this point to this point, or from this point to this point in millimeters, and then compare with the breakpoint on the standard before we can conclude that fosomycin is effective against this microorganism. So you measure for gentamicin, then you indicate the concentration. So if it's gentamicin into bracket 10 micrograms, then dash, you record is it sensitive or resistant or intermediate. Come to cephositin into bracket the concentration. If it's 15 micrograms, you record sensitive based on the breakpoint on the standard and what you measured. So when the clinician sees the results, then he or she will look at the ones that are sensitive. So assuming fosomycin is showing sensitivity, vancomycin, moxifloxacin, cotrimoxazole, and doxycycline. So 1, 2, 3, 4, 5 are giving sensitive outcomes. The clinician may decide to prescribe fosomycin or vancomycin. Remember, they will be targeting the same pathway. So he will not prescribe vancomycin and then fosomycin, but may prescribe fosomycin and maybe cotrimoxazole, or only cotrimoxazole or only fosomycin, depending on how wide the diameter is and the difference between this and then the breakpoint. So this is how they select the antibiotics for the patients or the clients for treatment. Hope it's clear. All right. So how are the results interpreted? We have the two category system where you compare with the breakpoint, you record the diameter in the diameter in millimeters, and then indicate sensitive or resistant. So if you measured, let's say 15, and the chart for that antibiotic, the standard breakpoint is 15. So 15 and above will be sensitive. Assuming you recorded 14, 14 is less than the 15, so you record as resistant. That means the clinician cannot prescribe this because it's resistant. He will be looking for the S, sensitive or susceptible. But this two category system is not recommended And the WHO has come up with a 3-category system where we record sensitive, resistant, or intermediate sensitive. Here, the intermediate sensitive is indicating that the organism is in between the sensitive and resistant buffer. So the clinician may prescribe the antibiotic that is showing intermediate sensitive, but with caution. He or she may have to increase the dosage, because at that concentration where you tested, the organism may become resistant with time. But if the concentration is increased, it will work and then become sensitive S. And the clinician will only fall on the I when there was no S recorded. So assuming for these antibiotics, you recorded R, 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 I, there's no S. Then the clinician has no option than to select any of the I's and then increase the concentration. I hope it's clear.
Yes, sir.
Yes, sir. But sir, I think they are not practicing the 3-category system.
They are not.
Yes, it's just the 2-category system that they do that. They don't do that.
They have to check. They have to check the standard. So these standards are updated regularly. They have the CLSI and then the UCAS system. So they have to be checking the standards that they are using for recording or reporting.
Thank you.
All right. Because if you are using the two-category system and there's no intermediate, you'll be recording those intermediate as sensitive. So the clinician will not know which ones to increase the dosage. And the patient may come back with the same condition. All right, so maybe you can check with them, and then in our next meeting, you can share with us. So you can ask them which year standards they are using.
Okay. Okay,
All right.
please.
And ask them which standard they are using. Is it the UCAST or the CLSI? All right, so antiparasitic drugs or medicines. So we have the antihelmintic and then the anti-protozoan. All these are parasites. For the antihelmintic, the worms that are able to invade the host, the benzimidazoles, example mebendazole, albendazole, they bind to beta-tubulin. These are proteins that maintain the structure or support the worms so that they can move, they can feed properly. So once these benzimidazoles bind to those beta-tubulins or structural proteins, they prevent formation of microtubules, which are very important for structural support. It's also very important in maintaining the organelles within these particular cells. So once the support is not there, they cannot pick up molecules. Movement also becomes a problem. And the organisms or the worms will starve and eventually die. And in some cases, they can also be used as anti-protozoans, antifungal agents, or even as agents against viruses. So remember, for some viral infections, even for COVID-19, they were using antibiotics for some of these treatments. So although they are anti-helminthic, they can also be used in some cases for other infectious agents. Praziquantel, also for the tapeworms and then the schistosoma species, and these are able to cause influx of calcium, and this will cause uncontrolled movement of those particular muscles within, or those particular cytoskeletons within those worms, and this depolarization will cause excess spasm or paralysis and affects the movement of these worms, and in effect, they cannot function properly. The ivermectin, the macrolides, so we have macrolides that are also antibiotics, but we have these that are anti-helminthics. They bind to the glutamate gated chloride channels, which serve to check the response to stimuli. So sending of signals to help the worms to respond to stimuli. So if they are starving and they need to feed, once these ivermectins bind to these channels, they make them unresponsive to these conditions, and then they end up dying. And this is what they use for onchocerciasis, strongyloidiasis infections. Then the nucleosamides and the tyrosantins, they inhibit energy formation by targeting the mitochondrial activities. You know, the mitochondria is the powerhouse of the cell. So once the electron transport chain is affected, there will be no oxidative phosphorylation and energy cannot be produced. Same for the tyrosantins that affect the RNA molecule production. RNA needs to be produced from DNA. So once this is halted, protein synthesis cannot continue. So the genes cannot be expressed. And if the genes are not expressed, the cell doesn't know when to multiply, when to feed, when to exchange molecules between the environment and the cell. They will also die. These are effective against schistosoma. So for schistosoma, they can use the tyrosantins, but commonly they use the praziquantel for mass drug administration. The anti-protozoans, we have the anti-metabolites, and there are several of them. So the malaria parasite is a protozoan, the Plasmodium, and atovaquone, proguanil. Usually they are combined for malaria prophylaxis, but they can also be used for treatment of babiosis, toxoplasmosis, and they target energy production within the parasites. Once they block the electron transport, it affects energy production, and then the cells cannot have enough energy for their metabolic activities. Proguanil also targets folic acid synthesis within the organisms. And you know, these are also eukaryotes, so they may also interfere with the same pathway within humans. So this is a challenge when we are taking certain medications that also target the eukaryotes. So if you increase the concentration, you may end up harming the host also. That's why we spread them for certain duration to achieve effective management. Artemisinin are also potent because they target different pathways. That's why they are very effective for malaria treatment or control. They target protein transports. They also disrupt mitochondrial function to generate more energy within the parasites. They also target DNA and RNA synthesis. They inhibit them so that replication cannot occur. And if there is no replication, there will be no cell division for reproduction to occur. So the numbers are reduced so that the immune system can take care of them. Then we have the sulfur drugs, the sulfadoxine, the sulfadiazine. So they combine this with the pyrimethamine for prophylaxis among the pregnant women. These also inhibit folic acid production. But unlike the proguanil, the sulfur drugs stay longer within the system compared to the proguanil. That's why they are preferred in pregnant mothers for protection. So we have those that also inhibit the synthesis of DNA and RNA. The nitroimidazoles, the quinolones, where you have chloroquine, amodiaquine, mefloquine. So for malaria treatment, they usually combine the artemisinin derivatives. So artemether, arteether, artesunate, all these derivatives, combination with the partner drugs such as the quinolones. So mefloquine, amodiaquine. Chloroquine used to be administered alone, but because of resistance, we moved from monotherapy to bitherapy. But these days, because we have stopped using chloroquine for over 20 years, there is a return of chloroquine sensitivity. And it's likely that the WHO may, at a point, recommend going back to chloroquine usage, but it also has to do with the side effects and all these things. So we are just waiting for WHO to give the green light. Metronidazole, tinidazole, these are the nitroimidazoles, and they are effective against the protozoans like Giardia, Lamblia, Entamoeba histolytica, Trichomonas vaginalis. Then the pentamidins, they are effective against the Trypanosoma and then the Leishmania species. So we come to the antifungal drugs or medicines. How do you know the difference between drug and medicine?
Sir, explain.
So the drug is the active chemical that is not packaged. So it works directly in the body. So if someone takes paracetamol is a drug. It works directly, so pain relief. But if it is packaged, that is medicine. So not all drugs are medicines, but all medicines are drugs because they are packaged.
Sir, please, if it's packaged as in.
So formulated. So some of the drugs need to be converted or metabolized before they can become active. In their raw form, they will not work. So some of them also need to be directed to particular sites. So in the formulation, they add other components. So it becomes packaged and that is what we refer to as a medicine. So for the human consumption, refer to it as medicine. But the actual active component is the drug. So not all drugs are medicines. Is it clear?
No,
Yes,
sir.
sir.
No, sir.
No, sir.
No, sir.
So,
Please, can you
No,
give us
sir.
examples so that we can understand it well?
so let's say this one. Let me use malaria, for example. Artemisinin. Dihydroartemisinin. Dihydroartemisinin is a drug, but we don't administer dihydroartemisinin directly. It needs to be metabolized from one form before we get the dihydroartemisinin within the system. So we have derivatives of the artemisinin. So artemether will become metabolized into dihydroartemisinin. So it is packaged in the form of artemether, which is a derivative of the artemisinin. So once it is packaged with other components, but the medicines are drugs because they are packaged so that they can be used efficiently within the system. There are certain drugs that can be taken directly, like paracetamol, for pain relief and all those. But not all drugs can be taken directly unless they are packaged with other components. Is it clear now?
Yes, sir.
All right.
Yes.
Are you doing pharmacology this semester or next semester or last semester?
Please,
Last semester.
Sir, last semester.
semester done.
Oh, okay.
Sir,
Yes,
please,
Serena.
we want an example of medicine.
I just gave one example. So same for amodiaquine. Amodiaquine is medicine. The active
Okay.
ingredient is not amodiaquine. It's decyl amodiaquine. But you cannot take the raw form of the active drug or compound. That's why it is formulated so that it can be taken within and then metabolized to get that particular active form. Is it clear?
Yes, sir.
Yes.
Sir,
Yes.
please, is it the same as some syrups that
Sorry?
are bitter? I said, is it the same as some syrups that are bitter that they have to add some sweetness to it before you can take?
So it's all part of the packaging.
Okay.
But the purpose is to make the drug get to its target. Some of them, if you take them in the raw form, they will not get to their target site. That's why they are formulated. Some of them in the raw form, they may be harmful, so they need to be formulated so that once they get to their target, then they can achieve their purpose. So the packaged drug is the medicine. All right. So somebody is asking, does it mean when you take SP, you have to avoid folic acid? Yes, but that's what you are doing in practice. Or do you give SP and then folic acid at the same time? No, right?
No, when we give the SP to them, we tell them to wait for 3 days before they start with the folic acid.
Of course, so that you reduce the concentration to a level before they start with the folic acid, because this one targets the folic acid synthesis. All right. So let's move on. We are on antifungal therapy. So the challenge I was talking about is because they're also eukaryotes. Please mute. Elizabeth. So Esther is now saying it's 2 weeks. Maybe because of the side effects. But once you take the SP stays in the system for at least 60 days, but the concentration gets reducing in the first week. And maybe that's why Esther is saying 2 weeks. Some of them will metabolize fast. Some of them also have whose genetic issues that will cause slow metabolism. Anyway. So fungi are eukaryotes, or they are eukaryotic organisms, and therefore the medicine that is prescribed should have lower therapeutic index. So you are saying indeed pharmacology. So what is therapeutic index? As for this, we were taught. So which one would you prescribe? A medicine with lower therapeutic index or higher therapeutic index? Hello?
Sir, please, the question again.
I first asked what is therapeutic index, and the second question is which one would you prescribe? A medicine with lower or higher therapeutic index?
Sir, higher therapeutic index.
Why?
Sir, because of the therapeutic effect it will have on the individual. The higher the index, the higher the therapeutic effect.
Are you sure? So therapeutic index deals with the safety as well as the expected efficacy outcome. So it's a ratio. So if you are having lower ratio, that means the medicine is working all right as expected, and the toxic level is also low. So it's also safe or tolerable for consumption. But if you have higher therapeutic index, the medicine is working all right, but the safety concerns is what you have to also look at. So for fungal treatment, the medicine should have lower therapeutic index so that you don't harm the one taking the medicine. The fact that you want to kill the fungi does not mean you should increase the concentration or the dosage, because fungi and humans are both eukaryotes, and we share a lot of biochemical pathways. Most of us, because we are all eukaryotic, we share most of these biochemical pathways. So the drug or the medicine that we take to kill the fungi may also be affecting our biochemical pathway. So if you don't reduce the dosage, the concentration, we may end up killing ourselves. So for these antifungal medicines, mostly we consider the differences in biochemical pathways, and we target those ones that we don't share with them so that we can effectively kill them. That's why it's difficult to get a lot of treatments for these fungal infections. And that's why we take most of these antifungal medicines for a long period of time at reduced concentration. So instead of taking high concentrations within a short time, we spread it and then we take in smaller doses. And these fungi are also very smart. They have a way of detoxifying the medicines or modifying them so that they become inactive within them. And that's how they also develop resistance to most of these medicines. And one key difference is the sterol synthesis. Humans, we have the cholesterol, but they have ergosterol. So we target their ergosterol synthesis pathway, and then we can kill them. So repeated dosages so that you can reach the concentration needed to kill or inhibit these ones effectively. So triazoles, imidazoles will target the same pathway, but the late stages of the ergosterol biosynthesis, while the allylamines will target the early stages of the ergosterol biosynthesis. So mechanism of action of these antifungal medicines, we have the imidazoles, the triazoles, the allylamines. So for the imidazoles, the miconazole, ketoconazole, cotrimazole, mostly for skin infections, vaginal yeast infections. Then for the fluconazole, itraconazole, primaconazole, the triazoles, they are for systemic yeast infections, the oral thrush, and then the cryptococcal meningitis. So the clinician may target this synthesis in the early stages, then go for the triazoles and the imidazoles in the late stages. So if it becomes chronic or recurrent, then usually they go for the triazoles and then the imidazoles. So Serena is asking how you measure the index. So pharmacology, you assess the safety level, then the level of efficacy, the level of tolerability. Then you have the ratio. But you have to also consider the various concentrations that you are using to measure. That's why for medication, we have the weight-based also method considered, a weight-based approach, because children may not be taking same as adults. So for the allylamines examples, the terbinafine, naftifine, for dermatophytes, the skin infections, so ringworm, jock itch, athlete's foot, and then fingernails or toenails infections. These can be used effectively against them. Then we have those that also target the same ergosterol, but here they disrupt the cell membrane by binding to glycerol. And example, we have the pollens, the amphotericin B, the nystatin, and these are mostly also for yeast infections of the skin, the mouth, and then the vagina, and for intestinal infections that are of fungal origin. Then the same cell wall synthesis for yeast, we can also use aspofungin for inhibition of the cell wall synthesis so that the cell will get exposed and then more molecules can move out and the cell becomes disrupted and eventually dies. So for the microtubules and cell division, glycophorin commonly used for dermatophyte infections of the skin. And these also target the proteins that will provide the structural support. And these proteins, once they are inhibited, they cannot support or pull apart the chromosomes for cell division to occur. So it prevents multiplication or reproduction of these fungi. But the challenge is because we take them in small doses, the time needed for the killing or the suppression to be achieved will be very long. And that's why most people take these antifungal medicines and they stop halfway because they are not seeing any improvement. You see the improvement when you continue to take them. But people get discouraged and then they stop halfway and they are saying it didn't work. It could have worked when you took high doses, but this could also harm or damage some of these organs. That's why they prescribe in smaller concentrations. Then we have the antimetabolites also affecting these metabolic activities. So we have the naptoquinone, the atobacone for treating pneumocystis pneumonia that will target the coenzymes that are related to energy production within the mitochondrion. Once these enzymes get inhibited by these antimetabolites, the transport of these electrons out of the matrix to create the right charge is inhibited. Please mute. Nakoko. So fluorocytosine inhibits protein synthesis. So if you are conversant with the DNA structure or the RNA structure, this is a modified nitrogenous base, and once they become modified, they cannot progress to being translated. So it means that the genes cannot be expressed beyond this point. And once the genes are not expressed, the fungi cannot have the living activities ongoing. Then the antiviral agents. So management and treatment of viral infections like HIV, infection, COVID-19. Fortunately, we don't have Ebola in our setting. But rabies, flu, chickenpox, we usually have such cases. And viral infections are also somehow difficult to control because they live within the cells of the host. They don't have cells. So as you try to inactivate them, you have to be strategic. Otherwise, you may be harming your own cell. They hijack your or the host cell machinery. So treating them, you may be targeting a pathway that will be inhibiting the processes within your own cells and not the virus. So to inhibit the nucleic acid synthesis of such viruses, drugs like CODeL are used for herpes viral infections or for varicella, the chickenpox, or the shingles, the Epstein-Barr virus infections, or the herpes virus human infections, and then the cytomegalovirus. This acyclovir also is similar to guanosine. So the structure of DNA is made up of 3 components. We have the sugar component, which is ribose or deoxyribose, which has a 5-carbon ring. It has phosphate groups and the nitrogenous base. So this acyclovir that is used for targeting the nucleic acid synthesis is similar to the guanosine. So guanosine is the guanine, which is a base bound to the sugar. So the structure for acyclovir, you see the 2 rings, purine ring, the same purine rings here. Then there is the 5-carbon sugar, the ribose sugar, which is lacking in the acyclovir. They have the open chain. So in the course of protein DNA synthesis or DNA molecule production, once these drugs are administered, the incorporation of the acyclovir replaces where there should have been guanosine. And because the structure is changed, there is no addition of another nucleotide to form the long DNA strand. This is where the nucleotides are added at this point, the 3' end. So this is the 1', the 2', this is the 3'. But because there is no ring here, there's nothing to add here. So it will halt or inhibit the synthesis of DNA or RNA. And once this is achieved, the virus cannot replicate. So this is how the acyclovir works to treat chickenpox, shingles, and the other viral infections. Then ribavirin, sofosbuvir, these are also used to target the nucleic acid synthesis. So for hepatitis C infection, respiratory syncytial virus, RSV, this ribavirin is also mimicking the structure of guanosine, just like we saw in acyclovir. And once this is incorporated instead of a guanosine, it also inhibits the production of DNA or RNA. This DNA is what the virus will use to replicate, or the RNA to take hold of the machinery and then be transcribed, translated into proteins. Then it can have the viral cycle completed and then exiting to infect a new cell. Same for the sofosbuvir, which is effective against hepatitis C. Here, it targets the RNA. So we have uridine in RNA and not DNA. So in place of uridine, this sofosbuvir also gets incorporated, and then the RNA polymerase cannot continue to transcribe the DNA into RNA. We have those that block the enzymatic activities, and several of them. Examples, we have the neuraminidase inhibitors. So oseltamivir, zanamivir, these are used for influenza viral infections. And remember the viral life cycle, at the last point, the virus needs to be released to infect new cells. But if these drugs inhibit the enzymes from releasing the virus, becomes self-limited within the infected cell, and they cannot spread to infect new cells. So the immune system will take over and then clear them. Then reverse transcriptase inhibitors, this is mostly for RNA viruses like the HIV, the COVID-19, or the coronavirus. So azidothymidine or zidovudine, these are nucleoside analog inhibitors. So when you talk of nucleoside, so a base bound to a sugar in RNA or DNA is a nucleoside. So in the case of the guanosine is a nucleoside because we have a sugar, pentose sugar, and then we have a nitrogenous base. So this is a nucleoside. But the technical name is guanosine because this is guanine bound to sugar, pentose sugar. When you say nucleotide is when you have a phosphate group attached to this 3' end, so it becomes a nucleotide. But nucleoside is when you have just the sugar and the base. So once you have these reverse transcriptase inhibitors in place, the RNA virus will need to convert its RNA. Once it penetrates the host cell, it needs to convert its RNA back to DNA before it can replicate. But if this is blocked, the RNA will stay in there and never get converted to DNA for replication to go on. So this is how they're able to end the viral replication process. Then we have those that block other enzymatic activities like protease, where we have the viral life cycle at a point where they unpackage to have the host cell mechanism hijacked, and then once they want to exit the cell, they need to repackage. But these medicines, ritonavir, they block the repackaging of the viral proteins and prevent them from getting assembled again to exit the host cell. And sometimes they can be used in combination to achieve effective action when the interferons are activated to clear viruses like the hepatitis C. Then integrase inhibitors, so as the name suggests, integration. So you have the HIV virus that needs to recombine with the host cell chromosome or host cell DNA. So this drug will inhibit the incorporation of the host, the viral DNA. So by this time, if the drug used was not reverse transcriptase to avoid or to inhibit the conversion of RNA to DNA, the virus may be able to convert it all right from RNA to DNA. But when it gets to this stage, this integrase inhibitor will prevent this HIV DNA virus this time from becoming incorporated into the host chromosome for replication to occur and then achieve the infection of other cells. But the viruses are also smart. They keep changing, and that's how they're able to avoid most of these medicines from working. And we have those that target the chemokine receptors. So fulveritide prevents the binding of HIV. No, for the viral life cycle, it needs to bind before it can penetrate and then unpackage. So these receptors on the host cell will be blocked so that this virus does not bind to the new host cells to infect them. So with time, they get cleared by the host system and the viral load will reduce. Then we have the fusion inhibitors, where this same type of antiviral medicines, the fulveritide, once they successfully bind to the host cell, they prevent them from penetrating. But before they penetrate, they need to remove their envelopes. So they'll prevent these envelopes from being released before they can penetrate or fuse with the host cell membrane. So once this is blocked, they stay on the outer part of the host cell membrane and they get cleared with time. All right, so that's about controlling microbial growth. We have looked at outside the host, sterilization, disinfection, and then within the host, chemotherapy or antimicrobial treatment. Any question up to this point? Yes, Millicent.
Yeah, please, it's not about the topic. It's about our examination. Please, we want to know the nature of the question. Will it be MCQs or there will be a section 2?
Do you want to know or you are talking for everybody? Because
Sir, we not me alone, we all of us.
so we'll talk about that next week.
Next week.
Sorry?
Okay, sir.
So we have the last lecture next week.
Okay.
But if you have any free period, we can have it either on Friday or Saturday. Otherwise, we meet at the same time.
Okay, sir. Thank you.
All right. Next week is not revision. If you check the academic calendar, revision is the week after. Do you know when you are starting the exams?
On the 5th.
When are you starting
Yes.
the exams?
On the 10th of April.
Well,
And today's date is 29th.
yes, please. That one is for practicals.
Who is saying practicals?
You know, for the level 4.
For practicals and exams.
My sister, Bisano. So practicals na nka ho a.
Mr. Munja Amaterewa, why? Munja Amaterewa, Muncha so and Amaterewa to more.
Show me nkasa you, Mr. Munyamia. Why? Sure.
All right, so see you next week. Have any question?
Okay, sir.
Right.
Okay, sir. Thank you.
Welcome.